For the sake of time, we cut here the Q&A and go to the next talk, which is Luis Diambra on predicting position of individually sequenced cells from their transcriptome data. Your stage.
Good afternoon. My name is Luis Diambra. I am very happy to be in this meeting, and I thank to the organizer for the opportunity to tell about some research obtained for the prediction of the position of the cell from single-cell transcriptomic data. As you know, the recent advance in single-cell technologies have driven a revolution in biological science in many senses. Despite that in Argentina we don't have enough financial support to perform single-cell transcriptomic experiments, I am particularly interested to keep in touch with this technique due to the potential to access many interesting questions in biology. The way that I found to keep in touch by this is by developing computational method for processing this type of data. This is my main motivation to be part of the Human Cell Atlas. My lab is a small multidisciplinary group dedicated to system biology. We are hosted in the Regional Center of Genomic Research, which depends on the National University of La Plata and CONICET. We are located in the city of La Plata, around 50 kilometers from Buenos Aires city. Going back to our topic, the single-cell transcriptomic approach allows the quantification of gene expression in many cells and explore the heterogeneity of complex tissue. Unfortunately, the information about the spatial position of the cells on the tissue is lost upon the dissociation of the cells from the tissue. One way to recover this spatial information is to properly combine the single-cell transcriptomic data with a reference atlas containing the expression patterns for several genes across the tissue. This approach was followed in several papers, like this. These papers emphasize the need for computational method that allows to reconstruct the spatial information lost during the dissociation process. In this sense, the past year, my lab participated in a dream challenge about the prediction of individual position. This challenge was organized by Dream Challenge together with IBM and Sachet Biot Network. My lab participated of this challenge with a group integrated with two postdocs, Andres and Alejandra. They came from biology and I'm from the physical science. I found interesting to tell our experience here, despite that this challenge don't deal with human single cells. The computational challenge consists in to explore for new methods to reconstruct the spatial information by combining the single-cell RNA-seq data with the reference atlas. In this case, the single-cell data correspond to the transcriptome of 1,297 individual cells from the fly embryo at stage 6. At this stage, the cells are located in a single layer with bilateral symmetry, so only position in one half of the embryo are needed. These include almost 300 locations or positional bins. The single-cell data is provided as a matrix with many genes for each single cell. Of course, many of these matrix elements are zero due to dropout and represent one of the main problems to deal with single-cell data. On the other hand, the atlas consists in 84 expression patterns obtained by in situ hybridization of the genes. These genes are regarded as determinant of the different tissue during Drosophila development, and some of them are organized in a regulatory network. The data for this challenge were published in this paper in the 17th. Despite that authors have been successful to reconstruct the expression pattern using a data-driven strategy, there are some open questions regarding the information used. For example, how many genes are needed for the reconstruction procedure? which are the most informative genes. And it is possible to devise algorithms that include other biological information. For example, one motivation of this challenge was the device method that include the regulatory link of the network that determine the embryonic patterning. I will tell briefly about the data in this challenge. In the pre-processing step, the in situ expression of 84 genes are binarized with a manually choosing threshold for each gene. And the single cell RNA-seq data are processed to obtain the gene counts relative to all genes in all individual cells, then are normalized at the pseudo count, and the logarithm of that is taken. Then the normalized data are also binarized using the same threshold value for all genes. The third step consists in to compute the Matthew correlation coefficient. This is accomplished by counting the number of genes that are true positive, true negative, and the number of mismatches. These numbers fit this formula for the Matthew coefficient. This is made for each positional beans and for each single cells. This coefficient can be organized in a matrix like this. The combinatorial expression of these 84 genes is enough to classify almost every position in the embryo without ambiguity. This plot shows the percentage of the cells assigned to a unique position versus the number of driver genes used to predict the position. As expected, the percentage of cells well classified increases with the number of genes in the reference atlas. The challenge asks for methods that predict the cell position by using a small number of genes, using only 60, 40, or 20 genes out of the 84 driver genes. So one important question is how to choose the genes for build the reference atlas. In our proposal, we select the genes to be used in each case by taking into account two criteria. Genes that have complementary expression patterns across the single cell population, and genes with expression levels broadly distributed across the single cell population. Regarding the first point, it is well known that there is some degree of consistency in the expression pattern. For that reason, we cluster the genes of the atlas with similar expression profile across the cells by using Euclidean distance over the normalized gene expression over the normalized gene expression levels, and the unweighted per group method as a linkage method. Then we cut the dendrogram 3 into 20, 40, or 60 groups depending on the case. In this way, one can identify clusters of the genes with complementary expression pattern. The second criterion is accomplished by choosing in each cluster that gene which maximizes the entropy. In this plot, the arrow shows the performance in the cell position prediction obtained using 20 genes selected with this criteria, while the yellow distribution is the performance obtained when the genes are randomly selected. It is interesting to remark that among the main motivation for this challenge was the possibility to having an advantage with the knowledge of the gene regulatory network. However, when observing the results obtained by different teams that participate in the challenge, this knowledge don't seem to be an evident advantage. In this graph, we can see that the most of the teams selected very few driver genes, which determine the cementation process in the fly. Notice that the genes are sorted taking into account the frequency in all cases. The pink bars correspond to the case with that use only 20 genes. We can see that there are not any driver genes among the 10 most frequently selected genes. We see that from the binarized atlas of selected genes and the binarized data from single cells, one can compute the Matthew coefficient. To reconstruct the spatial profile of any gene, we can use this coefficient and the single cell expression levels. The expression levels of a given gene G in a positional bin P is the summation of the expression level of the 10 better scored single cell weight, weighted with the Matthew coefficient, as shown here. Notice that in this strategy, an individual cell can contribute to more than one positional bin. Finally, I want to close the talk with a contribution to this strategy that consists in using the reconstructed spatial pattern of expression to expand the number of genes used as reference atlas. We can compute again the new Matthew coefficient with the extended set of genes, then predict the position of single cell, and reconstruct the patterns again in an iterative manner. This is particularly valuable when the atlas consists in a small set of genes. In this plot, we can see the performance of the prediction using a set of 20 genes. There is an improvement at each iteration, and after 3 steps, we have around a 10% of extra efficiency. We have noticed that this iterative procedure is no longer efficient after 3 or 4 iterations. This plot illustrates the reconstruction expression pattern of Fuji-Taratsu genes, a driver gene involved in flight segmentation. This reconstruction was obtained using only 20 genes after 3 iteration steps. We can see the typical 7-bundle structure of these payroll genes. Each sphere corresponds with the positional bin, and the intensity of the color is associated with the expression level. The gray spheres correspond to positional bin without assigned expression level. Well, that is all. Thank you very much for your attention.
Thanks a lot, Luis. Great talk. Maybe I can kick off with one question, which is a little bit off topic. Could you tell us a little bit more about these dream challenges and how those are organized and how people interact? Do you have like pre-contact with other teams that work on the same topic and exchange experience, or you just present a final result and then you benchmark one against the other technique?
Well, my first idea was present some results in the for initiated contact with other groups in order to access data and more experienced people to teach me about technologies and method, computational method for to perform this.
Okay, and you applied your approach on a very like defined, organized setting. So can you, for like other like biological clinical scenarios, do you think?
Yes,
if the systems. Sorry, yeah, go ahead.
I am interested into not specifically into predict position. I am interested mainly into reconstruct networks from trajectories, but we are in the first steps about that mission. So for this time, this is the only research that we have, and only preliminary for single cells.
Okay, and do you foresee like problems when you work on maybe less organized tissues or even disease tissues or cancer, including microenvironments? Do you think an approach like this would work, or you would face like major problems there?
Oh, I don't sure if I understand the question. Can you repeat me, please? For
some tissues have a very organized structure. Some
yes,
others are less organized. And
like
in
this,
disease, the picture might look different. So I guess it might be hard to predict in this context.
Yes, I have no idea about that at the moment, but I think that in this case about Drosophila is the case of a very simple tissue without any structure or complex structures. So maybe was a good case to a good tissue to make to ask the question about what are the more informative genes, how many genes you need to reconstruct the position or the patterns.
Yeah, I agree. It's probably the best data set for such challenge.
Yeah.
Thanks a lot, Luis.
Thanks.